Good morning, everybody. I hope you are having a good day. This next presentation will be lecture number 2 on the lead optimization methods that we have been discussing for the past few weeks. So just a background again, you guys can go through all of the background, everything that we've been doing so far. In my previous lecture, we spoke about lead modification methods, the identification of the pharmacophore, functional group modifications, SARs, and the use of privileged structures. So now what we will do is go through what type of modifications we can actually do to these compounds to either increase their potency, their therapeutic index, or also to modify their ADME properties. So what is the goal of structural modification? The main goal is to increase the therapeutic index of the drug. You can maybe also include here, it's not just the therapeutic index, but also to modify the ADME properties of a compound. But a therapeutic index is a function of the dose which gives the maximal therapeutic effect in 50% of experimental animals, that is known as ED50, and the dose which kills 50% of experimental animals. So the lethal dose that kills 50% of rats or mice or whatever animal you are using in your experiment. Then they use this equation where they take the lethal dose over the effective dose, and they do a calculation. So what we want with lead modification is we want the lethal dose to be large. What that means is that it needs to be at a concentration, let's say 100 micromolar, which is a very, very high concentration where the drug becomes lethal. But we want the effective dose to be very small. And let's say, for instance, we have a dose that is effective at 0.01 micromolar. That means that your therapeutic index will be very large. So that is what we are aiming for here, to have a drug that is very effective at a very low concentration and that the lethal dose is very high. So for instance, if you have that drug that even if you take, like, for instance, 30 tablets or so of the drug, it will still not lead to the lethal dose concentration, just as an example. So that is why we look at therapeutic index as well when we do structural modifications. And remember, we can still do structural modifications once we get into animal studies and animal testing. If you do see that there is a problem, that you get to toxic effects or lethal doses in animals, then you can go back to the drawing board and try and optimize your compounds so that they are not so toxic or lethal. And it does happen, and we do use this data then to do further structural modifications and structural improvements. So the therapeutic index, there are certain diseases which are serious diseases where a therapeutic index is low, where there is a relatively low lethal dose that is close to the effective dose. And there's two diseases such as cancer and AIDS, where it is acceptable for a low therapeutic index. So the reason why this is acceptable for cancer and AIDS is because we don't have better therapies for cancer and AIDS. It's still an unmet medical need. There's still ongoing research to find more improved therapeutic options for these diseases. But currently what we have, we know of chemotherapy and how sick chemotherapy can make you. But that is the best that we have now. If we look at the risk versus the benefit outweighs the risk currently for the treatments that we are using for cancer and AIDS. But there's definitely an unmet medical need, and many companies, pharmaceutical companies and research groups at universities globally is still working at improving therapeutic agents for both cancer and AIDS, and hopefully also with a much larger therapeutic index, so that we can treat these diseases much more effectively. Let's have a look at some of the structural modification techniques that are used in drug design and discovery. The first one, which you would have seen also in the additional lectures that you were supposed to watch with the transcripts. Most of these techniques were already discussed, but I would just like to give you some overview and my opinions on this as well. So the first one is homologation, where homologation is basically where a molecule differs by a constant unit, and it's usually a CH2 group. So if we, for instance, look at this example here, this molecule, it's measured in its potency. So the more potent the drug is, the higher this value will be. And here you have your chain length. So it's from a 1 carbon chain length to a 2 carbon, 3 carbon, and so on, until you get to a 8, 9, 10, 11 linker carbon at this R position. So if we look at this graph, you will see if that R position is substituted only with a CH3, a methyl group, we see that this drug is not potent at all. We don't see any effect. But as we start increasing that chain length, doing homologation, 2 carbon, 3 carbon, 4 carbon, 5 carbon, 6 carbon, once we get to a 6 carbon chain length here, we see that this molecule is very potent. If we then increase the carbon length further to a 7 carbon, 8 carbon, and then to a 9 carbon, you see here at the 9 carbon, you have abolished the activity completely. So what do we observe in this graph? What could be the possible explanation? And we need to look at the explanation from both a pharmacokinetic and pharmacodynamic viewpoint. So if we look at the pharmacodynamics, which is the effect of the drug on the body. So it is the pharmacological effect of the drug on in the body. As we can see here, as the chain length increases, we get very good activity. And that may be because this drug with its biological target, it interacts with its biological target. That chain length is optimal for the drug to fit into that biological target, span maybe that entire active site, and show the optimal potency. And if you then start increasing the chain length again, what happens? The drug becomes too big for the biological target, for the active site, and it won't bind to that active site, or not as optimally if you look at the 7 carbon chain length versus the 6 carbon chain length. And if you get to an 8 carbon chain length, the drug is just too big and it won't fit to that biological target, and you will see a massive drop in activity. So that may be a reason for the pharmacodynamic explanation of why we are seeing this effect on this graph. If we look at pharmacokinetics, if we look at the ADME properties, so as this chain length increases, you get an optimal chain length with optimal lipophilicity, optimal Lipinski's rules of 5. And this drug is then great for absorption, great for metabolism, and it is still effective. But then what happens if you then start and increasing that chain length more? The drug becomes more lipophilic. And obviously, there will be an issue with delivery of the drug, because if a drug is too lipophilic, it may not cross biological membranes. It may just get stuck in a biological membrane, because it's a fatty substance, and a biological membrane is also a fatty substance. So it then just gets dissolved if the lipophilicity is too high. And that may be the reason why we are seeing this massive drop as the chain length increases, which will lead to an increase in lipid solubility as well. And if we look at the other side of this graph, why do you think that if we only have 1 or 2 carbons up to 5 carbons, we don't see as good activity from a pharmacokinetic standpoint? So for pharmacokinetics, that may mean that the drug is not lipophilic enough to cross biological membranes. So it may be too hydrophilic. Hydrophilic meaning it's too water soluble. Highly water soluble drugs will not cross biological membranes. So that may be another reason why we see with shorter chain lengths with this drug, why we don't see the drug actually reaching its site of action, and obviously then potency will be affected. So you see there's a very close balance between what you're looking for from a pharmacodynamic point, so the activity, versus the pharmacokinetics, which is ADME. There needs to be a balance. You can't just have a highly potent drug with poor ADME properties. You also can't have an effective drug that has great ADME properties, but it has low potency. So you're always in your drug design projects, and if you design a drug design project, there always needs to be a balance between the activity and ADME, and this graph explains it quite nicely. I did put it into words in the next slide, so you can go through it in your own time, and you can also watch the additional lectures and transcripts for this section. Then very similar actually to homologation is chain branching. It is basically just a different way of doing homologation. Like for instance, if you look at this drug over here, promethazine, you can see there's a branched CH3 group, so that's chain branching. Here you don't have a branched chain, and with trimethazine, you can see you have the additional branched chain as well as homologation here. So they've actually increased the number of carbons in the straight line, as well as they increase the number of carbons by including a branched CH3 group. And you can see by just doing these very simple structural modifications on these phenothiazine type drugs, you can see that there's a massive difference in biological activity. And the reason for that is basically with that small chain branching difference, they bind to different receptors. Because maybe this one with that branched chain, there's something happening there that does not able to make it able for the drug to actually get into the active site of that target, but may be appropriate for another target. This one over here, you can see there's no branched chain here, so this drug is slightly smaller. It is slightly more linear. So this one may then be able to reach a different target compared to promethazine. And with the biological tests that they've done, they've seen that chain branching may change the biological activity quite drastically of these phenothiazine drugs because they bind to different receptors. And you can look at some of the biological effects that was observed and what researchers then saw when they did the biological evaluations on these three drugs. Now, you can also do ring chain transformations, which is also very similar to homologation and chain branching. Here it's just adding more carbons and closing ring structures. And obviously that will also then lead to a different way in which the drugs will bind to its biological target. And obviously in this case as well, it may also affect the ADME properties of your drug. So in some cases, if it does affect the ADME properties, the drug may not even reach its site of action. So let's say, for instance, the one drug you want to have it to work in the central nervous system, and you use chain branching to add on to the number of carbons to make it more lipophilic, then obviously the drug will be able to cross the blood-brain barrier and work on the CNS. Whereas in other cases where you maybe don't do chain branching, it's not lipophilic enough to cross the blood-brain barrier, and it won't work in on the central nervous system. So that is strategies that they use to make drugs also more site-specific for a biological effect. So ring chain transformation, chain branching, and homologation is old strategies that we use in drug design and discovery, but it's still applicable today, because if you go through your structure-activity relationships, if you build on your structure-activity relationships, you can use all of these techniques and then optimize your molecule by looking at the biological effects and ADME properties and the differences thereof. Then bioisosteres. I'm not going to spend too much time on bioisosteres. You know that you can use that Swiss bioisostere server to learn a little bit more about bioisosterism and also to see what type of bioisosteres you can see. So for this section, take note that there are certain functional groups that you can change, and the activity of the molecule will relatively be retained as well. So, for instance, in this case, we have CH2 groups. CH2 groups can be also substituted with an NH group, an O, S, or selenium. Here you have a carbonyl, that's just a CH2. That can be substituted by an amide, an ester, or this type of compound. So I don't want you guys to go and memorize all of these bioisosteres. And so what I want you to know is just that bioisosteres are groups of compounds that have similar biological activities and that you can use in drug design and discovery projects. And you also get some other examples as well. For instance, if you have here an aromatic group, aromatic groups can also be substituted with all of these other aromatic groups. You can use this as your strategy. Check what is the ADME properties, check what its biological activity then is. Does it change the biological activity? Does it increase therapeutic index? So bioisosterism is a strategy that you can use to build on your number of compounds and then build on your structure activity relationships in order to then make better informed decisions of which type of molecules you need to go forward with. So, for instance, I just given you this one example of bioisosterism. So in this case, it's again the phenothiazine drugs, the normal phenothiazine drugs. There's the sulfur bridging group at this position. And if you look at non-classical bioisosteres, and please, you guys don't have to know about classical, non-classical bioisosteres. Just take note that there are two different types. But in this case, it's a non-classical bioisostere, that's sulfur, and it can be substituted with a double bond or a single bond because it has a similar size, shape, and electronic nature compared to this sulfur molecule here, but it doesn't have the same number of heavy atoms, so making it a non-classical bioisostere. So here you can see we have these three different molecules, but we also then see that we have differences in the biological effects of these three compounds. Why do we see some differences in biological or pharmacological activities when we do bioisosteric replacement? Now remember, bioisosteres are similar functional groups. They are supposed to retain the pharmacological activity. Now remember, retain the pharmacological activity. They don't say if a bioisostere may affect the potency. So you may have a bioisosteric substitution where you still see the pharmacological effect, but a much higher potency. You may also do a bioisosteric replacement. You still retain the pharmacological effect, but you may have a drop, a massive drop in that pharmacological activity, but that drug can then still be described having that biological activity, but it's just lower. So bioisosteres does not keep the exact same pharmacological effect. There's still differences where higher or lower potencies may be observed, but the pharmacological class of the drug will remain the same. So the reason why we do see some fluctuations in the pharmacological activity when you do bioisosteric replacements is the size of the compound changes. So the compound may be too big, so then you don't see as good pharmacological activity because it can't bind as effectively to its biological target. The shape of the compound may differ. Again, then it may affect the way in which it binds to the active site. It may affect the binding interactions of the molecule. The electronic structure of the compound may change. Even though a bioisosteric replacement is just on that one functional group, it may affect the electronic nature of the entire compound. And the electronic nature of the compound may also play a role in its ability to reach the site of action, because highly ionized, negatively charged compounds does have a problem with absorption as well. So they may not be able to cross a biological membrane if you do change the electronic structure of the compound too much, which will lead to a drop in potency because it won't reach its site of action. Then also bioisosterism may change lipid and water solubility. They may make the compound too lipid soluble, so it can't reach its site of action, or they make it too water soluble. Then it doesn't cross a biological membrane because highly water soluble compounds can't cross biological membranes. And then also the way in which it binds to its biological target. We spoke about that. With bioisosteres, the size and shape may change, and it may then affect the binding to your biological target. So those are some of the reasons why bioisosteres may then lead to slight drops in activity or slight increases in activity. So it's a very nice strategy to use to quickly and easily build structure activity relationships for you to then study and then make informed decisions on what is your next step in your drug design project. Now, peptidomimetics, I'm not going to go through peptidomimetics. You have the transcripts and videos on peptidomimetics as well. We will get back to peptidomimetics next semester. For now, just take note of what a peptide is, what a peptidomimetic is, and how we use that in drug design and discovery, and have a look at the transcripts as well that is on our eCanva site for this section, and make a few notes on this section. There will probably just be like 1 or 2 MCQ questions in exams and tests on peptidomimetics, and we will come back to that at the later stage. Then, very important part as well, we do structural modifications also to increase oral bioavailability, because oral drugs are always the type of drug that we are trying to develop in a drug design project. It's because of patient compliance and the ease of taking an oral drug. It's much easier to take an oral drug than for instance, an intramuscular drug or an IV drug. So we always try and develop oral medication, especially for diseases such as HIV, malaria, all of those diseases that we see here in South Africa and Africa. Obviously, you won't be able to always use an IV drug if you go to rural parts of South Africa and Africa. It will be a problem because who is going to then administer an IV drug? And if that IV drug needs to be administered weekly or maybe even daily, it becomes a problem. So we are always striving for, in drug design and discovery, when we're working with small molecules, which we are doing here, molecules with a molecular weight of less than 500 grams per mole. We are trying always to get to an oral bioavailable drug. So just some statistics. So 75% of drug candidates do not go to clinical trials because of pharmacokinetic problems. We spoke about this previously. I think it was in the first face-to-face lecture where we spoke about this. And then less than 10% of drugs then make it into go to clinical trials. And 40% of the molecules that fail in clinical trials do so because of pharmacokinetic problems. And this is the reason why we've been using that Swiss ADME server. That is a very nice tool where we can then already get a prediction of what the pharmacokinetics will be like for your molecule. So if you do see that there is a problem with the pharmacokinetics, at least you can get that that information very, very early in your drug design project. If you work for a company, you can use Swiss ADME and you can tell the company, listen here, we shouldn't go for this drug. We can already see that there will be a problem with its lipophilicity, there will be a problem with its solubility, and so on. So we can, with drug design now, we can early in this in a drug design project already make an informed decision on potential pharmacokinetic problems. So we are moving towards a way in which we can make it much better and get less drugs to fail during clinical trials. Because you must remember, if a drug fails during a clinical trial, that pharmaceutical company loses millions and millions of US dollars. They don't want to fail during clinical trials. And they are developing these type of softwares to make it better, to make it more efficient, to make it cheaper, and also then to hopefully lower the number of drugs that go into clinical trials and then fail, because that is a massive expense globally on the failure of drugs. And the moment that we can have less drugs that fail during clinical trials, drugs and medicine will become cheaper as well, because you don't have to then, the pharmaceutical company can then charge less for that medicine because they don't have to cover their losses if the ADME problems is identified quite early. So it is very, very important to make sure that once you get to a drug that goes into clinical trials, that you make sure that you have a good chance of success with ADME. You guys can go through all of this as well. I have mentioned this continuously throughout the course so far. Then how do you measure lipophilicity? Lipophilicity is basically that balance of the drug to tell us if it's more lipid soluble or if it's more water soluble. So you need a balance between water solubility and lipid solubility for a drug to cross a biological membrane. If it's too water soluble, it doesn't cross the biological membrane, no chance. If it's too fat soluble, lipid soluble, then as we've already discussed, it just then basically gets stuck, it gets dissolved in the membrane and it doesn't cross. So you always need that balance to have optimal lipophilicity for the drug then to cross its biological membrane and reach its site of action. Now we can measure this experimentally in the lab. And how we do this is we simulate a biological membrane by using an organic solvent known as 1-octanol, which simulates the membrane. It is a highly fatty type of organic solvent. Even if you get it onto your hands and so, you struggle to wash it off with water. And you will feel also like a fatty layer forming on your hands. I have experience with this because I did these type of studies during my master's degree as well. In those days, we couldn't do ADME predictions. There were no software available for us to do ADME predictions. So we still had to go through this experimental part where we use 1-octanol and water. And then the water stimulates the cytoplasm. The more drug that's in the cytoplasm, the more water soluble it is. The more drug that's in the 1-octanol phase, the more lipid soluble the drug is. So you would then add your drug to the let's explain it a little bit differently. So what you would do first is you would add 1-octanol and water into a flask. You would shake that flask, and then water and octanol can't mix with one another. So you get two layers. At the top, you will see the octanol layer, and at the bottom, you will see a water layer because they are not miscible or not mixable with one another. You would then add your drug of interest to this mixture. You would vigorously then shake the mixture and let it stand for half an hour or so. What you will do then, you will take a sample from the 1-octanol and you will take a sample from the water phase. You would take those two samples and maybe use a technique such as HPLC or UV absorbance, and you will measure the amount of drug in the octanol and the amount of drug in water. And then you use this equation here. You guys don't have to know how to use this equation. I won't ask a question on this. But then with this equation, it gives then us a measure of lipophilicity known as the partition coefficient P between octanol and water. So luckily these days, and luckily for you guys, we don't really have to do these experiments anymore in the lab. We can now use online tools and online software such as Swiss ADME to do the predictions for us. And you will see that Swiss ADME, if you check it experimentally as well, Swiss ADME is pretty accurate in their prediction of log P. Now, you see a few equations and stuff here. Don't worry about the equations. What I want you to know here is the larger the log P value, the partition coefficient value, the more interactions with of the drug with the membrane. So the more lipophilic the drug will be. Larger log P equals more lipophilic. For a drug, and this is very important, for a drug that you want to develop for a CNS condition, it needs to have a log P value of more than 2, but less than 5. So you can maybe make a note of this. So optimal CNS penetration for a lipophilic drug, where a log P needs to be between 2 and 5. If it's less than 2, the drug is not lipophilic enough to cross the blood-brain barrier. If it's more than 5, the log P, then the drug becomes too lipid soluble and it will just get stuck in that blood-brain barrier, that biological membrane, the blood-brain barrier that basically divides our bodies into the CNS and peripheral. So that is the range for optimal CNS drugs. So if you want a drug that not to work in on the CNS, like for instance, cardiovascular drugs, then there's no reason for you to go and design a compound with a log P of more than 2. Because if you do have then a compound with a log P of more than 2, that will lead to an increase in CNS penetration, and it may lead to an increase in CNS side effects if you have a cardiovascular drug, and that would not be a good thing. So once, for instance, if you make a cardiovascular drug, you will always try and keep that log P value less than 2 to ensure that you don't get CNS side effects of that drug as well. So that is just a strategy that they sometimes use if you don't want to see CNS side effects. But unfortunately, many drugs have been made, for instance, for cardiovascular diseases and for other diseases that don't affect the CNS, but a small amount of those drugs still cross the blood-brain barrier, gets into your CNS, and that's probably the reason why sometimes you get effects of side effects of certain drugs, like, for instance, nausea and vomiting, headaches, and all of those kind of things. And it's probably because there's a fraction of the drug that still crosses the blood-brain barrier. So this is a strategy that you can use if you keep it below a log P of 2 to still enable your drugs not to cross the blood-brain barrier too much, and then lower the side effect profile of your compound. And obviously, if you want the drug to cross the blood-brain barrier, then the log P needs to be between 2 and 5. CNS diseases such as Alzheimer's, Parkinson's, Macedo-Joseph disease, stroke, all of those kind of things would need drugs that have log P values between 2 and 5 in order to cross the blood-brain barrier. Now, a tool that we use to determine whether or not a drug will be oral bioavailable. You probably already saw this in your first practical when we did Swiss ADME practical, and this is known as Lipinski's rule of 5. So Lipinski's rule of 5 states that poor oral absorption, and make a note of this, the way that I've set up this slide is for poor oral absorption and or distribution is more likely when the molecular weight of the compound is more than 500. Then you will probably see some poor absorption. If the log P is is above 5, then you see poor oral absorption. If there are more than 5 hydrogen bond acceptors, so if you take hydrogen bond acceptor, it's basically all of those functional groups that are OH and NH group. If you add all of the OH and NH groups together, and if those number of groups are more than 5, then you have a problem, because then you may also then see poor oral absorption. If you then go and you add up all of the N and O atoms within your chemical structure, and all of them are then more than 10, then also you may see some poor oral absorption. You can maybe also just make a note for hydrogen bond acceptors. That is actually the sum of all N groups, all O groups, including OH, NH, and NH2 groups. So it's N, O, OH, NH, and NH2 are all hydrogen bond acceptors. Hydrogen bond donors is only the sum of OH and NH groups within your structure. If you just look at the name, H bond donor, so it needs to donate a hydrogen. And obviously with OH and NH, there's a hydrogen group here that can donate that hydrogen. For N and O groups and so, they can accept a hydrogen group. So it is highly likely, more than 90%, that compounds with 2 or more of these characteristics will have poor oral absorption. There are exceptions to this rule, like for instance, the antibiotics, antifungals, vitamins, cardiac glycosides, and even the drug levodopa that we discussed at length during our stereochemistry last year are exceptions because they may use active transporters to cross biological membranes. So this is also something that you need to keep in mind when you do a drug design project, that you shouldn't always just look at the lipophilic nature of a compound. Sometimes you also have to look at active transport mechanisms, because you can maybe use that as a strategy to get drugs across biological membranes. Now, the next slide is just an example of a drug that is dissolved in water. A very simple example. So you see, this is the drug, and then you have dotted lines here that reacts with water molecules. Now, if we look at the number of hydrogen bond donors and the number of hydrogen bond acceptors within that compound, if we first look hydrogen bond donors, the number of OH and NH groups. So here I see an OH group. And here as well, I see an NH. This is actually an NH2. NH2 two groups you can maybe add in here as well is also a hydrogen bond donor. So please include NH2 here as well as a hydrogen bond donor. Here we have the NH2, and here we have the OH. So we have two hydrogen bond donors, which is good. The molecular weight of this compound is less than 500. Its log P is less than 5. And if we then look at the number of hydrogen bond acceptors, so that is the sum of all N, O, OH, NH, and NH2 groups. So if we add all of that together, here we have an OH group, there we have an O group, there we have an NH group. So all of these are then added up as hydrogen bond acceptors. So we have 3 hydrogen bond acceptors and we have 2 hydrogen bond donors. Now you will see for this OH group can act as a hydrogen bond donor and as a hydrogen bond acceptor. So we can see here OH, the functional group, interacts with a hydrogen bond donating interaction with water. With the OH as well, that O group of the OH can form an interaction with the hydrogen of water acting as a hydrogen bond acceptor. When you just have the O group, you see that there's no hydrogen bond here or hydrogen group here, but we see that there's still an interaction with water where this carbonyl group, this O group here, can accept that hydrogen, forming that hydrogen bond interaction. And a similar thing happens here with this NH2, where the N group can form a hydrogen bond acceptor interaction, and the NH then form a hydrogen bond donor interaction. So my question to you now is, do you think that this molecule will be water soluble? Yes, this molecule will be water soluble because there's numerous interactions with the water molecules, making it soluble in water. And it has a log P value that is less than 5. You don't see too many hydrogen bond acceptors and donors. It still abides to Lipinski's rule of 5. So this drug will be orally bioavailable. It will be soluble in the gastrointestinal tract, and it will in all probability be still able to cross biological membranes. Now, the last few slides is just a case study, something that I did a few years back. It's actually a study that's published as well, where we identified the number of privileged structures that could be used for Parkinson's disease. Now, you can look at each of these chemical compounds and have a look at their log P values. Do you note a problem with these compounds? Because they are for Parkinson's disease, so we need the log P value to be between 2 and 5, right? So if we look at all of these compounds, we can see that it's only this one here, and this one here with a log P value of more than 2. But that for me is too close for comfort. That is basically at 2. So I would like to see that value a little bit higher to give us a better chance of better ADME properties. And obviously these others, especially this one here, has a log P of 0.51. So even though it's a privileged structure that can be used to design agents against Parkinson's disease, this compound will never cross the blood-brain barrier because it is just not lipophilic enough. It's actually quite water soluble. So we then devised a strategy where we combined this structure here. This structure is known as amantadine. It is already a FDA approved and a SAPRA approved drug for the treatment of Parkinson's disease. It blocks a receptor known as the NMDA receptor, and it reduces excessive calcium flux within cells. So excessive calcium influx in in brain cells leads to a process known as excitotoxicity. And excitotoxicity then leads to oxidative stress, which is basically like, think about how you peroxide your hair. If you peroxide your hair, it becomes white. So the same thing that happens with this is that when you see oxidative stress, it happens to your neurons as well. Your neurons get stressed by these free radicals, by oxidants, and then or they also get like peroxide, what you see with your hair, and they die off. So amantadine can stop that excitotoxicity to a certain level to slow down the progression of Parkinson's disease. So the strategy that we try to use here is to use this amantadine already that is FDA approved and then conjugate it to all of these different molecules to see if we then can improve the ADME by increasing the log P to between 2 and 5 to have a good ability to cross the blood-brain barrier, and then also then to increase the therapeutic index and therapeutic effect by having this adamantane structure that's already effective against Parkinson's disease in slowing down the degrees. It's not a cure, it just slows down the disease, and then combining it with one of these privileged structures, and then hopefully get a much more improved molecule. And we actually did it. We went into the lab, we did the chemical synthesis, we tested the ADME properties, and this is what we came up with. And you can clearly see here, when we combined the adamantane structure with our privileged molecules, we got log P values that were much more better compared to what we previously saw. We even got 2 compounds with log P values that were a little bit too high. But then what we found is this molecule here, that is the adamantane conjugated to the coumarin structure. This is known as a coumarin. And it shows a log P of 3.5, which is right in the middle of optimal lipophilicity for CNS penetration. And this molecule also showed quite a few good neurobiological effects, which could make it a much better therapeutic agent for Parkinson's disease. And we've been working on this compound, I think now for about 7 years, on and off, and we are still in the process of further exploring it to see if there is potential for this drug to go a little bit further into preclinical trials and hopefully one day even into a clinical study. So that just shows you some strategies that we can use in lead modification to improve the therapeutic index of your compounds. Okay, thank you, ladies and gentlemen. I do hope that you've enjoyed this presentation, and good luck for the rest of the week, and keep well. Thank you.